My name is Corinne Stobaugh, and I'm a volunteer with KSF. I use my background as a pathologist and a Kabuki mom to 11-year-old Gabe to help KSF with bridging the patient and research communities. Last week, I was listening to all of the amazing research endeavors from the scientific symposium, and it got me thinking about one of my absolute favorite things, jigsaw puzzles. Specifically, how forming a clear picture of how Kabuki syndrome works is like trying to put together a 10,000 piece puzzle with tiny pieces that are all shades of gray. It's just a vastly complex disease with dozens of different mutations and two of the largest genes in the nucleus, and expressed nearly everywhere DNA is translated in the human body. Just to put that complexity into perspective, some of the genetic diseases that I learned about in medical school have just a single point mutation in a single gene that is expressed in a single tissue type. But despite the complexity, as was apparent in the scientific symposium last week, there are groups of researchers all around the world working diligently to not only put their own pieces of the puzzle together, but to help others put theirs together as well. To take that progress even further, as we have learned this morning, the study of biomarkers through the KSOC program is an essential part of the Kabuki syndrome puzzle that needs further study and collaboration between our patient and research communities. To help us do that, our expert panel of KSOC researchers is here to answer questions at this virtual fireside chat. So let's welcome Dr. Rosanna Weksberg from the University of Toronto, Dr. Margaret Adam from the University of Washington, Dr. Evdokia Anagnostou from Holland Bloorview Research Institute, and Dr. Jackie Harris from the Kennedy Krieger Institute. Thank you all for joining us. So let's get started. Can each of you let us know your academic background and briefly describe your research? Maybe Dr. Weksberg, you can start us off.
Sure. So I'm a clinician and researcher at the Hospital for Sick Children in Toronto. Can you still see me?
Yep.
Okay, I just got booted out of something, sorry. And my research work is on disorders like Kabuki, where the proteins involved really regulate normal developmental processes. So what these proteins do is they go along the DNA and turn on and off certain genes in specific tissues at certain times to make sure that all development proceeds typically. So when there's a problem in the Kabuki syndrome gene like KMT2D, the tags that go on the DNA to turn things on or off at certain times aren't properly placed. And so across the genome, across the DNA, you get a difference in the tag placements. And this is what we measure when we measure DNA methylation. And it is a specific pattern that we get. That's my clinic that was supposed to not call me. Anyway, we get a specific pattern that we can identify as Kabuki specific. And what we have found, and the reason we're going into this KSOC trial, is that you can actually get specific patterns associated with specific phenotypes or certain features of Kabuki. So we're hoping that we can correlate the neurocognitive issues with DNA methylation biomarkers, with the hope in future that we can use it in a clinical trial and potentially even predict early on which individuals would benefit from a treatment. That's kind of a shortcut version.
Dr. Adam, can you also let us know your academic background and briefly describe your research?
Yes, so I am also a clinical geneticist, and my interest in Kabuki syndrome really lays with understanding the clinical features and helping other researchers to, similar to Dr. Weksberg, come up with best practices for monitoring individuals with this condition, with the goal of also helping to bring a potential therapy at some point to the community. So most of my research has focused on trying to delineate what are the clinical features, how many people have each type of feature, so that we can really understand the whole spectrum of what we might see clinically in a person with this condition, that would then help us understand how we can care for them in the best way possible.
Thank you. Dr. Anagnostou, can you tell us a little bit about your academic background and describe your research?
For sure. Hi, everybody. I'm a child neurologist by training, and I'm a clinician and a researcher here at University of Toronto at the Bloorview Research Institute. And most of my research is trying to understand how the differences in our genetic code, the program that actually decides how we build proteins and how we build bodies and brains, affect the brain and the body, and how then these changes in the brain and the body affect the way we think, our behavior, what we're really good at, and what we maybe struggle with. And we do this across different genetic syndromes and other neurodevelopmental conditions where we don't even know what the original genetic difference is, to try to find all the different ways that kids who have these types of differences are different from each other, and how we can personalize. The hope is that we can then personalize the interventions to the biology of the child that's in front of us, not just some average child that has a label, but the child that's right in front of us, to make sure that the treatments that we offer have the best potential benefit for the risk that every treatment has and every family has to take.
Thank you. Next, Dr. Harris. A lot of us are familiar with you, but can you also let us know a little bit more about your academic background and describe your research, in case anyone else is joining us?
I am a neurologist, a child neurologist also, and a neurodevelopmental specialist at Kennedy, Krieger, and Johns Hopkins in Baltimore. And so I study the same group of disorders that Dr. Weksberg studies. So we call them epigenetic disorders. Only I, like Kabuki syndrome, only I study them on the clinical trial implementation end. So actually getting clinical trial readiness and then actually carrying out those clinical trials. So what do we need for good clinical trials in these disorders? So starting with looking really, really in depth at these disorders, like what Dr. Adam was talking about, like what are all the features of the disorders and how do we know about them? And then how can we better understand mechanistically the disorders, like Dr. Weksberg does, to develop biomarkers and outcome measures like we were talking about. And then like Dr. Anagnostou was saying, also developing these sort of protocols for testing therapies and treatments, and making sure that we do this in a way that we get the best possible answers to the question of, do these things help, and get them to patients quickly if they do.
Thank you. Do any of you have any comments, additional comments, because you kind of went over it in your research, but how having the biomarkers validated by KSOC program affect your own personal research and the understanding of Kabuki syndrome as a whole? We kind of went over that a little bit, but if you have any additional comments, let us know.
Yeah, I mean, I'll just quickly say that the biomarker process for the FDA, as well as all the other kind of FDA-like bodies in other countries, is pretty extensive of what you need to actually be considered a biomarker in a specific syndrome for a clinical trial. And so these kinds of studies are incredibly important. They really, really advance the science, and they make it easier for any therapy that you bring about to have ways to test that therapy. So it's not just specific to one drug, one diet, one treatment, but actually looking broadly at how can we measure any of these that come about.
Dr. Anagnostou, do you have any comments about how the importance of biomarkers will help your research and the understanding of Kabuki syndrome?
Yeah, I mean, the way to think about biomarkers is that they tell us something on top of what we know from having Kabuki syndrome. And so parents know that if they look at everybody's kids in a conference when they get together, although they all have Kabuki syndrome, they look different. And some of them may have different needs, and some of them may need different interventions. And so what the biomarkers hopefully will do for us, they will tell us which version of Kabuki syndrome a child has, so that the intervention is matching to the version that this child has and not just to the whole category of Kabuki syndrome.
Thank you. Dr. Adam, do you have any additional comments about the importance of biomarkers?
Yes, so kind of really going based on what we've already heard, for a clinician, understanding whether an intervention is actually being helpful to someone is not always obvious on a clinical side. So the changes we see may be too subtle for some of our tools that we currently have in clinical practice to be able to detect. And so this is, again, another tool to help us understand how is this impacting this person's care, and how can we follow that over time.
Thank you. And Dr. Weksberg,
So
can you,
one of the
please.
big draws for me in terms of biomarkers is that, as people have mentioned, Kabuki syndrome children are different, and they can look different because of the way they process the variant that they have, but they also bring to the table other things that they have inherited from their parents or from the environment. And so one of the things biomarkers can do is to make sure that you're bringing into the trial individuals who are actually having a deficit in the differences that we want to target. So you may have a variant in the Kabuki syndrome gene that we're not sure about, and the biomarker will tell you whether it actually is related to what we're going to be looking at or not. So it actually makes the children that go in initially much more homogeneous and much more likely to give us an answer, and reduce the heterogeneity or the variation in the population that goes into the trial.
Great. Thank you so much. So I just want to remind everybody watching that there is a Q&A section, and please enter in your questions, because we will be having a Q&A session after this. So as you're thinking, as the researchers are talking about all those very interesting topics and about biomarkers, please add your Q&A questions, and we will get to them after this session, the Fireside chat. So I'm going to ask the researchers a few more questions before we get to that. And let's start with Dr. Weksberg again. Why do we need so many people to participate in the study? Clara said there was about 90.
Right. So as I was saying just a few minutes ago, there is a lot of variation. And some of that is just population variation. And so we have to make sure that we get rid of that variation and are really looking at the variation related to Kabuki syndrome. And in order to do that, we need that number of participants so that we can actually make sure that we're finding differences that are the true differences that we want to follow and capture in a biomarker.
Okay. Dr. Anagnostou, what exactly does the KSF funding pay for?
So the funding will pay for two buckets of things. One bucket is the cost of the families trying to participate. And so we want to make it as easy for families to participate as possible. So the foundation is very generous at trying to pay reasonable costs for families to get to the site to actually contribute their time and their samples. The other bucket is the people that need to do the work to collect the information. So some salaries for psychologists and technicians that will be collecting the information, but also the stuff, right? The tubes, the liquids, the things that need to go into the samples so that we get good quality samples. So that's basically what the foundation is paying for, to kind of facilitate us getting this research started.
Thank you. Dr. Weksberg, how will you ensure a diverse set of patients?
So we are going to track the entry of patients in terms of age, sex, country of origin, ethnicity, and we're going to make sure that, and it's really important when you develop biomarkers, that you actually reduce all of those variables again and just stick to the differences that you really want to focus on in developing the biomarker. So that's the critical piece.
And also, if anybody has any comments about a question, please feel free to chime in.
Oh, can I just add one thing? There is actually
absolutely.
a very generous travel possibility in terms of money, so that we can actually access people not only in North America, but internationally.
Okay. Dr. Adam, are there risks associated with participating in the study?
So there's always a risk in any study that people participate in. This study has kind of two different arms to it. There's the clinical piece and then the sample collection. The samples that we're wanting to collect in this study are primarily blood and urine samples. And so the risk for having those types of samples collected are very low, the same risk that you have anytime that you have a blood draw, which most people have had numerous times. And so the risks on that side are pretty low. In terms of the clinical side of things, there certainly are always possibilities that we're doing some pretty extensive neuropsychiatric evaluations, and some of the questions that we ask or results we get may be uncomfortable for people as they're going through. We will have some questionnaires as well, and some people may feel uncomfortable answering certain questions, although the participation is completely voluntary. And although we would hope that people would be able to answer every question, there's no requirement that every question has to be answered in that scenario. And of course, there will be clinicians there to help support the family and the participant.
Thank you. Dr. Anagnostou, if a child is nervous about going to the clinic or doing blood draws, but still wants to help, can they participate?
Absolutely. So we, first of all, no family has to do something they don't want to do. But we also know that very few children will just put their arm out and say, take my blood. So we know it's a procedure that's tough for a lot of kids. And sometimes they just need support and facilitation to get through it. So we have lots and lots of techniques to make kids comfortable, to distract them, to give them things that they like, to use numbing cream, to try at different times, to work with the family to make it as least invasive and as least distracting to the kids as possible. And honestly, sometimes part of the gain of the study is not the original intent of a study, which of course we want to learn a lot about Kabuki syndrome to help kids with Kabuki syndrome. But at an individual level, sometimes going successfully through a blood procedure, when in the past it has always been a struggle, is kind of an empowering experience for kids that they take with them after, like an experience of success. So we will try with all the kids, no matter what their discomfort level, to walk them through, to make it more possible for them, more pleasant, and a successful experience. So we want to try. If you're willing to try, we will try with you.
Great. Dr. Harris. Sorry.
Can I add one thing? Is that
Absolutely.
all of us and all of our teams, this is what we do. We work with kids with neurodevelopmental differences and behavioral challenges, and just kids in general. And so if there is anything that a family is concerned about, has a question about, nervous about anything, but they, are interested in participating, please don't hesitate to reach out to us or our study teams. We are happy to answer those questions prior to, making any arrangements to visit, so that families can feel more comfortable in understanding that any part, just like Dr. Anagnostou said, any part to participate in is your choice. We don't make families do anything they don't want, but we also facilitate maximal comfort so that anybody can participate in everything that they would want to.
Yeah, we heard from the previous participants that you guys have a great child life program as well. I guess continuing, Dr. Harris, can we send samples to you instead of coming to the clinic for the KSOC program?
So while there are other research studies in which you could send samples to us that we could use for other things, for KSOC specifically, we can't just use samples that you send to us. The reason is not because there's anything wrong with those samples specifically, but more so because the point of this study is to actually validate these things as biomarkers. And as such, we really need the extensive cognitive and behavioral testing and kind of medical understanding of your child that we're gaining through these visits that we wouldn't have access to if we just had a sample sent to us on a child we'd never seen before.
What about old samples? What if we've been to another facility within the consortium and had samples taken before?
It would kind of strongly depend on how those samples were processed. But again, the key would be that we need to correlate any samples with, the active, what we call phenotype, which is the active symptoms and presentation and cognitive testing of that person.
Can I
Thank you.
add to that?
Oh, please.
Okay, so I just want to say that DNA methylation does change over age. And so there are things that we have to make sure are connected so that our assumptions are correct when we do all the analyses. So I would say that we really do need these in real time. I mean, if it was a week before and it was a sample that was available, then yes, of course. But that's the kind of thing we're doing.
Okay. Dr. Adam, will participants be able to see their results and the results of the study?
Most of what we're collecting is, as has been said by my colleagues, is to evaluate the utility of certain kind of findings to be used for biomarkers. And so much of what we're collecting on the biological side is really, we don't understand how that would be useful for clinical care now. And so that's what we're trying to investigate. In terms of the cognitive and neurobehavioral testing, we are not planning on automatically providing that to families, but families could request that information, and then the team will kind of go through and decide what we are going to tell the family. I can let Dr. Harris talk a little bit about the EEG findings. And then we hope that with this project, we will be able to publish in a peer-reviewed paper the biomarker profiles and how we think they might be useful. And that would be kind of a summary across all participants. But again, at this point in time, we don't really know if these are going to be clinically useful or how they are going to be clinically useful.
Right.
Yeah, so for EEG, if there's anything that we see on an EEG that would be clinically relevant to a patient, then we would definitely let that patient know that they need to go get further evaluated with a clinical EEG. But the EEGs that we're doing are completely research-based. They don't have any by themselves clinical value. It would only be if, for instance, in the very unlikely scenario, hasn't happened yet with an individual with Kabuki syndrome, but in the very unlikely scenario that we saw a seizure on an EEG in a participant who had no idea that they were experiencing seizures, that of course we would have that further evaluated and disclose that. But as far as the actual data we're collecting for the EEG, it's not clinically useful. And then as far as just like Dr. Adams said, the neuropsych testing is available on request. So our site makes all of that available. That's your data. You can have it, but it's important to note that the only thing that we generate from that is scores, right? So you would just get literal scores printed off. It wouldn't be like if anybody's ever had a clinical neuropsych eval where you get these, big 14 pages of descriptions with recommendations and here's how to change your IEP and things like that. We are not able to provide that on a research basis because this is not a clinical neuropsych eval, but we certainly provide scores so that families can take them back to their own clinical teams and things like that and do what they can with that.
Great, thank you. Dr. Adam, how long is the KSOC opportunity open for? If you can't participate in 2024, what about 2025?
Yeah, so it's going to depend a little bit. So we are hoping the study will run between 2023 and 2025, meaning that participants would be enrolled during that time frame. But it also depends a little bit on kind of which centers have how many patients and how many patients have enrolled already. And so there will be a little bit of dynamics to that, but people should be able to hopefully be able to enroll in 2025 as well.
Of course, we hope we get 90 participants right now by people, filling in the form to the left right this very second. But on to the next question. Dr. Anagnostou, will the samples be collected for KSOC be used for future studies?
Yes. So when you go through your consent process, you will be, depending on the site that you're in, you may be told or you may be asked separately whether the samples that you have contributed can actually be banked, which basically means saved in appropriate conditions, so people can ask questions to them later on. And the kinds of things we usually think about when we bank samples like this are things that new procedures and new technologies and new money sometimes that would allow us to ask questions about how a genetic we know the gene, but how the many differences on this gene may contribute to differences in kids with Kabuki syndrome. We may be able to use more advanced technologies like whole genome sequencing, where we're not focused on one gene, but we're looking at all the genome of a child to understand how it contributes to the kinds of things that they're really good at, but also the things they struggle with. And ultimately, sometimes what happens is we develop what we call cell lines. So that's one way of preserving our ability to look at DNA, because many cells get created from the child, not only the original cells that we had in the original samples. So we can access that DNA, we can access that computer code that tells us how the body and the brain is being built for years to come, to ask new questions as we get better at our understanding and as our technology gets better.
Great. Dr. Weksberg, do you need sibling or data, or can family members come and donate?
So different sites may do this differently, but we certainly can use siblings in the broader study. We won't be using them in the initial analysis, but we invite you to discuss that with your study coordinator.
Okay. And then a question for all of you. What do you wish patients and families would know about getting involved in research, and how has your relationships with patients and families really inspired you in your own work? Let's start with Dr. Harris.
Yeah, so what I wish, I think the participants who you heard from a minute ago are probably better able to speak to this than any of us. But what I wish that families would know is just that I think that it can often be really beneficial to both families and the participants themselves to participate in research. A lot of the kids and adults with Kabuki syndrome that I see after they participate in research, they actually, feel pretty good about like feeling like they've contributed and they've helped and they've, helped a lot of other people, and that can be really, really beneficial. It also is, I think all of us at our sites we try to make it a good day. Like we have a lot of data to collect and things, but we like you heard, we have child life and we have, gardens and therapy rooms and playrooms and things where we try to make it a beneficial day that everyone will enjoy. You can come, you could extend your visits since we're paying for travel funds. If you want to pay for a little extra hotel time on your own, you can come visit any of our lovely cities or the surrounding areas. And I think it'll be good. And then for me personally, interacting with patients and families on these research visits, this is what I do with my whole life, right? Like everything I do is this. And so clearly it's extremely rewarding to me, and I love it, and it's where I get my energy from. And so I hope that it is beneficial to you all too. But take home points here, like we really need you. It's not just a statement. It's not just a thing we say. We literally can't do any of this stuff without you all.
Thank you. Yes, your enthusiasm definitely shows. So we definitely appreciate it. I want to ask the same question to everybody else in the panel as well. Dr. Weksberg, do you want to go next?
Sure. So I also see a lot of children with differences in my clinic, and I am often very excited to be able to share information that we generate through our research lab with families to help them refine their understanding of what the genetic differences mean, and also to use the data from multiple individuals to really help the entire community. And I want to echo Jackie's sentiment that we need all of you. Like, really, the more participation we get, the better data we will have, and the better chance we will have of really delivering something that we want to deliver to the community.
Thank you. Dr. Anagnostou?
Hard to add to this, but the way I think about this is for you guys participating in research, it should not be seen as a privilege. It's our privilege to have you, to see your stories, to meet your families, to meet the kids. But for you, it's a right, and you should see it as a right. You should demand access to research, because it is your right to have the decisions that get made, the therapies that get developed to reflect your child and your family's biology experience, individuality, individual differences, right? So we need you all because we need to capture all of these individual differences, and you should demand that we give access to all, because we need to make sure that anything we develop actually is useful to our whole community, and not a select group of families that happen to live really close by or find it particularly easy to have access. So we're incredibly grateful. It's a privilege for me. I spend, again, all my day interacting with mostly research patients, and but for you, it's your right. It's your right to have your individual biology and experience represented in the data sets that will be used for decision making that affect your families. So partnership, critical. I see you guys as our co-investigators, as our collaborators. This is meaningless. If we do it without you, it's meaningless. It's a waste of the trust that has been given to us and the money that has been invested in the space.
Thank you so much. Dr. Adam?
I have to say, it is hard to go last after all of that. But for me personally, because of especially my geographical location at the University of Washington, where we're a regional medical center, and there are not a lot of genetics groups that are in our region, many of the study participants that I work with, not only in Kabuki syndrome, but with other research endeavors that I do with other conditions, it allows me to understand more in depth what is really going on with a particular patient and with their family. And that I think really helps in clinical care overall. But I think it also gives me, and I think many families, the satisfaction of knowing that we're contributing to a larger community, to the overlying knowledge that we're trying to gather that gives us kind of a larger purpose with what we're doing, and being able to collaborate oftentimes around the world to bring together both clinicians and families, and to really have the opportunity to understand sometimes what rare complications might happen, the things that we might not be able to see if each one of us was seeing patients in a vacuum, to bring that knowledge together to share with the entire community.
Oh, thank you so much. Yes, we are definitely stronger together.
Corinne, can I say one quick thing?
Absolutely.
Which is that I really love what Evdokia was saying about how it is, not your, like, we are not twisting your arm into participating in research. This is absolutely your right to do and feel comfortable and choose as you will. And I will also, speak for everyone on this fireside chat, because I know everyone on this fireside chat, we are all exquisitely sensitive to negative connotations that the word research has had to certain communities and certain groups of people and all of those things like that. And so please, if there are any concerns, there is no question that you could ask that would offend or embarrass any of us. So anything that you want to say, can I talk to some more people who have participated, who look like me, who come from the same area as me, who any of these things, please, let us know, because we are here to facilitate your ability to choose to participate or not.
Thank you. So now we're going to actually move to our Q&A and bring in our KSOC participants, Erica, Avery, Misha, and Jimmy, who I have a bunch of questions, and let me just see if I can get to them. Let's see here. All right. Oh. Well, here we go. Oh, we also want to thank Dr. Bodamer of the Roya Kabuki Program at Boston Children's. Boston is the 4th clinic location, and we're so thankful for his expertise. He couldn't join us today due to family illness, but sends his warmest regards. So thank you again, Dr. Bodamer, and we definitely look forward to having the Roya Kabuki Program at Boston Children's as one of the recruitment places for the KSOC study. All right. So let me get to our questions. Okay, so these are being populated as we speak. So I'm just going to go ahead and start. Are all of our participants ready to go?
Yes.
Oh, all right, great. Thank you, Jimmy. So I have a question. Our son is nonverbal/autistic and 11 years old. Does this affect his eligibility for the study?
No.
No, I don't. Dr. Harris?
No, it doesn't. We will ask you some questions beforehand so that we can appropriately, know which parts of the cognitive battery are or are not appropriate for your child, but no, it doesn't affect your ability to participate. Love to have you.
All right. Does KSF cover the international flight fee to participate in the KSOC?
All of the sites have a travel budget for different participants. We have a certain amount of funds that are kind of acceptable. And then after that, we definitely work with families who have excessive costs to get them to a research site. And so the answer is probably yes. Please reach out to your research coordinator. I do, again, want to reiterate something that Clara said earlier, which is that right now, We hope to have this not be the case in the future, but right now for this first KSOC initiative, we need all the children kind of to be native speakers or native listeners in English. So that's the one international kind of caveat.
Okay. Here's another question. There's a variety of presentations in Kabuki syndrome. How difficult will it be to correlate the biomarkers with different presentations and the different genetic mutations?
Maybe I can jump in on this one.
Yeah.
And then somebody else can also jump in, because that is the million-dollar question, right? So we know that there are many presentations. We know that there are many different ways the gene can be different in the case of Kabuki syndrome. And we know that there are many different ways that sometimes the same difference can get translated into the proteins that are the building blocks of our bodies and brains. So there is a lot of different ways an original genetic difference can impact what behaviors and thinking skills and physical manifestations we see in a kid or an adult with Kabuki syndrome. So part of the call to get as many of you as possible is to capture as much of that variability as possible, so that we can use advanced mathematical techniques to try to understand how the biology difference and the presentation difference map to each other. If we don't have a lot of variability, we make assumptions. We want to make as few assumptions as possible. We want to have the actual data that will allow us to mathematically predict that what we see in the kids from the biology. But others can jump in.
All right, next question. So a lot of our participants mentioned that the blood draw seems to be an issue. How much are they taking? Like how many tubes and how much volume per tube?
I will just
So
say
I
that
can
it actually wasn't a lot because we spoke about it, but I think it's just the tension, the buildup of doing it and us being transparent with Avery. But go ahead, someone who is professional here can answer it.
Yeah, so I go with the participant for the blood draw, and I kind of just go on case by case. Like if you're just able to give, like if we can get like 4 tubes, and we don't fill these tubes to the brim, by the way. If we can get like 1 or 2 cc's, that's like wonderful. But if you're a hard stick, and if it's sometimes in a just smaller vein participant, we just prioritize our DNA methylation, and then we just call it a day. Like what we can get, we will take. And if we don't get anything, that's also great. We can try a different time. But it definitely is not a lot of blood. It's just getting the blood.
Thank you. Next question. My 14-year-old son doesn't have the gene mutation, but has every characteristic of Kabuki syndrome. For example, prominent ears, failure to thrive, fondness of music, high arch palate. Would he be a candidate for a research participant?
So I'll take that question. So we do have a number of individuals who fall into that category. And when we do the DNA methylation, some of them actually fall into a positive category. That is, they have the tags that we normally see with Kabuki syndrome. And so those individuals would be eligible, and we would do more work to actually try to find the genetic change. Some will be negative, and we have found a number of other conditions that actually overlap with Kabuki syndrome. And looking at DNA methylation signatures for these other conditions may actually raise the possibility of an alternate diagnosis that could be followed up clinically.
Mm-hmm. All right, next question. My daughter had neuropsychological testing last summer. Would she have to wait a certain amount of time before participating in this research opportunity?
great question. So when if your daughter were to enroll, we would ask for any recent neuropsych testing. And if it were the exact same test that we're running in the battery and done very close to when we would do it, we would just not do those specific tests and use the previous scores. But most of the time, it's not the exact tests because we're doing kind of a very specific, kind of Kabuki specific battery. And honestly, if it's anything a year or more back, we're going to repeat it anyways. So the answer is possibly we're going to ask you for your stuff when you enroll, and then we'll figure it out from there.
Okay. This question is for Avery. Someone has asked, I usually struggle with needles, so I would love to ask Avery what they did to help her.
You remember?
Kind of. I got to bring my iPad.
Which is a good distraction. And I think you had a stuffy. I don't know if it was.
What about Bluey?
It might have been. It might have been Bluey.
Nice.
And we did. I mean, we always depend on child life. And the nursing staff are great and gentle and really kind with you. And I was there. So as many people as needed, and distractions. And I believe I also brought gummy worms.
Oh, yes, gummy worms. That'll do it.
Jimmy, do you want to quickly say from your experience, give the participants some advice for blood draw? What helped you on the second day that you didn't do on the first day that gave us a better blood draw?
I drank a lot of water. Yes. Yes. Water is one of the main things that you need when you're doing blood tests.
Especially after a long flight from Australia to the US, right?
Yes. In saying that, I had been in the US for about 2, nearly 3 weeks at that point.
Oh, okay.
Yeah.
So you had some time to acclimate.
Yeah.
All right, next question. As the DNA methylations change over time, might the blood samples need to be repeated as the youngsters or young adults get older, which then can be compared?
So that's an excellent question. We look for the tags that remain there over time for diagnostic purposes. We may look for different groups of tags, depending on certain neuropsych testing. So we don't have the answer yet. It's one of the things we're looking at, is the change over time.
Here's another question. We visited the Roya Kabuki program last summer. Do I select that site as a preferred location, or is there value to visiting a different site for the KSOC program?
You should go wherever is most convenient for your family.
Do adults need to go somewhere other than the sick kids institutions for KSOC involvement?
I'm assuming this is a Canadian family, right? So the family will not go to Sick Kids. The family will come to Holland, Bloorview, which is uptown from Sick Kids, where we see all the kids with neurodevelopmental differences, and we have the ability to test you and provide the appropriate accommodations. But your blood samples then will get to Sick Kids at Dr. Weksberg's lab.
And there will be...
You can send us also direct emails to the teams, and we can walk you through the process.
And
there
when
will be
in 20... Oh,
I'm sorry. There will be an optional clinical genetics visit at Sick Kids that will be determined. Evdokia and I still have to connect about that. So,
but Canada adults are fine, right? I think that was part of the question too.
Yeah.
Yes.
Okay.
And when in 2024 will the Toronto sites commence seeing participants?
So I think we are making a prediction here, because what we still need for the Canadian side is the ethics approval and all the right contracts. So we are expecting it's the second quarter. So sometime end of March, early April, we should be able to get the families in.
Okay, and is the study focused on Kabuki 1 and Kabuki 2, or does it matter which one?
We'll take both.
Okay. Would having stem cells and cord blood tissue help with any research now or in the future?
Not
So,
for
like,
this
as
part
in-
of the study, but yes, the answer
Okay.
is yes.
Okay. So not part of KSOC, but yes for things in the future. All right. Is there research already going on into the connective tissue issues, which are part of the hypermobility problems, like we heard before about dislocated patella?
So, I mean, we'll, as part of KSOC, we'd collect your information about, you know, the joint issues that you've been having and things like that. And, you know, there are some studies currently published and some ongoing, centered mostly in the Netherlands, out of like using growth hormone to address some of the joint hypermobility issues. But right now, it's not a specific part of KSOC, but we will collect that information just as part of all the general medical information that we would collect.
And do you see that at a certain age? Hypermobility?
We see it at all ages and in different degrees in different people with Kabuki syndrome. Specifically the patellar dislocation tends to be more common, you know, it tends to be less common in the tiniest kids and a little more common as you get older, which, you know, mainly has to do with like growth and stuff like that.
Another question. My daughter is 36 years old and she works. Can any of the KSOC testing be done on Saturdays to minimize her time away from work?
Yes. Yes, at Kennedy Krieger, we are working hard, not right this minute, not November 7th, 2023, but by January 2024, we're working to move things all to research spaces away from clinical spaces so that those kinds of things can be a possibility.
Would it be only at the Kennedy Krieger location that Saturdays are available?
I mean, I don't know where this family is coming from. So if we had a family we needed to accommodate a Saturday, we would accommodate a Saturday.
Okay.
That's the same for the Seattle site.
Okay. How many of the target 90 participants are signed up so far?
As in signed up, meaning completed their KSOC, or as an expressed interest? Because they're a little bit different. So we've had an enormous amount of people express interest, but they haven't necessarily completed their visit. The only participants who have completed their visits are at the Kennedy Krieger site, because we were kind of the soft launch test site. And we have I might get this wrong, Ludwin, 12 completed, 11, I don't know. Ludwin, can you answer for me?
Yeah, we have about, that's about how much we have completed.
I think 11.
Okay. Next question. We saw Dr. Harris a couple years ago and did all of this testing. Would we need to come again, or can all that data be used for this study?
So we used that data, we published it, we used it to develop this study. All of that data was invaluable in making this study possible and knowing what to use. But no, we need the data again. If you can't come, depending on what study you participate in, we will use every piece of data that we possibly can use, but ideally we need new data.
Okay. Dr. Weksberg, can you explain the difference between chromatin methylation and DNA methylation, or are they the same? And which is more important?
Okay, so actually, these tags occur. So chromatin actually consists of DNA and proteins, which are called histones. So there are tags on both the histones and the DNA. And on DNA, you get methylation, which we assay. On histones, there are a large number of different kinds of tags. In order to look at the tags on the histones, you need live tissue and antibodies, and it's quite a complicated procedure. DNA methylation, however, can be assayed on stored DNA and is much more stable. So that's what we're doing the assays on. But we can correlate the DNA methylation output with what happens on histones most of the time. So when we go into the iPSC cells or the stem cells, we will be doing both kinds of assays because we will be working on cells. But for this study, we'll be working on DNA methylation.
Okay. Are participants able to extend their stay to make a vacation out of the visit? It sounds like, Jimmy, you were able to extend your visit in the US.
Yes, I was. We traveled for about 3 weeks. Yes.
You may want to post your itinerary for other people to share.
Yes, true.
Was it your first time in the US?
Yes, it was. Yes, we
Did you
went to
enjoy it?
Disneyland. We went to Universal Studios. We actually did a Kabuki camp, the gathering in Texas. So we took part of that, and we met other adults, and there are about 5 adults and a number of children with Kabuki syndrome with their families. Then we traveled to New York, and we did a few days in New York before coming to Baltimore.
Sounds like
And
fun.
we did Baltimore for a few days where I did the test, and then we had the weekend. I caught up with another friend of mine in Baltimore, and then we flew home on the Tuesday.
That sounds like a lot of fun.
Yes.
A way to make the most out of your visit.
Yes.
All right. Is it possible or in any way valuable to contribute to this or any other research based on the charted medical history of a deceased person with Kabuki?
That would be amazing. I am sure there are benefits that charted medical history can bring us in some capacity. For this initiative, we can't, because we can't get their DNA methylation at the point that we can look at all those records from. But yes, in the future, please keep in touch with us. Please talk to us because we would love to, have you and the deceased person be able to contribute in every way possible.
And if there is a bank DNA sample in a lab that performed the sequencing to find the Kabuki change, that would also be valuable and could be used.
Also, what a really beautiful offer.
So we still have a few minutes left, and we can, expand the Q&A to non-KSOC questions. Please continue to submit those questions in the Q&A, and we can continue until the end of the hour. Oh, here's one. Are there any plans for a KSOC study in Europe? We'd love to contribute, but travel from the UK feels like a lot.
Yes, we'd love to. There are some considerations we need to work out with, different ethics agencies and data sharing rules and language issues with the cognitive testing, but you know, that would be the eventual goal.
Next question. How can all of the data you collect from KSOC contribute to future research in Kabuki syndrome?
That's a wonderful question. So all of the data that we collect in KSOC, de-identified, right? Because we don't want any of your data to go anywhere that it shouldn't go, but de-identified data, we are making sure that it is available and shared in a de-identified form so that anybody who says, hey, I want to look at this, I want to look at that, based on the data, the clinical data that we collect, the actual prospective evaluations that we do, or any of the you know, biobank samples that we get, we absolutely want to share those for anyone who can use them.
We have another question. How old is the oldest person diagnosed with Kabuki syndrome?
The oldest person I've ever seen is in their 50s. I don't know if anybody's seen anyone older.
Next question. I'm interested in understanding the MTHFR side of things and whether having Kabuki syndrome puts patients at risk for other illnesses such as cancer.
So I think we don't know is the long answer. This is something that I've been interested in for a long time, because when you look at the underlying molecular differences in people with Kabuki syndrome, there is a concern that cancer could be a risk, but I would say clinically we haven't really seen that, and that happens in a number of different genetic conditions where we predict we should see something and we don't. But this is data that I think we don't actually have enough information on, and particularly in adults with this condition. And it's something I think that most of us are interested in trying to understand. But I think part of the difficulty is finding individuals with this condition who are adults and following them long term. And that's been challenging, partly because the genetic testing and genetic causes have only more recently been identified. And so people diagnosed many, many years ago in the 80s, some of those people probably had related conditions that weren't Kabuki syndrome, but we couldn't tease those out in a manner that was, you know, scientific.
How long will it take to complete KSOC, including data analysis?
So this first part will take a couple years. So, you know, 2.5 years or so, including data analysis. But our hope is that it's not 2.5 years. Our hope is that it's a little bit longer, because, you know, this first step is to really develop the biomarkers and get them sort of ready for what they would be. And then the second part would be to get in another cohort of individuals and kind of validate these biomarkers, or make sure that it looks the same in a different cohort of individuals. So whoever asked about, you know, are we going to miss the participation window? I highly doubt it, right? If you want to participate, if you have Kabuki syndrome and you want to participate in KSOC, I can promise you, I don't think we're going to be turning you away, because we definitely are going to have this first cohort and later cohorts to validate. So somewhere in the range of several years.
Okay. Can you explain more about Kabuki 1 versus Kabuki 2? And
there are two different genes that have related functions. One puts the mark on, the other one takes the mark off. The other really distinguishing feature is the Kabuki 2 is caused by KDM6A, which occurs on an X chromosome. So it actually manifests differently in females and males, which is not true for Kabuki 1, which manifests pretty much the same in both sexes. So, and there are a lot of things that need to be taken into account when you're working with Kabuki 2. So it will be easier for us to focus first on KMT2D because working with the X chromosome issue will complicate things for a number of reasons, both clinically and in terms of the methylation. But we are bringing both cohorts through. We just think we'll get data more quickly for the KMT2D group. Jackie, I don't know if you want to add to that.
Oh, agree. And KS2 is also just frankly far less common than KS1. So another reason why the analysis piece of things will take longer. Not that we're not, willing to, excited to see the KS2 individuals, just that it'll take us a little bit longer to get the numbers that we need to do all the things that we want to do.
Yeah. So again, like both KS1 and KS2 are able to participate. So please, whichever mutation your loved one has, or if you have it yourself, you can still fill out the KSOC form to the left of your screen. And because today is our launch day and we are really excited to hear from you. If there are questions you have that weren't answered, or you think of some questions later, please email us at KSOC, that's KSOC at kabukisyndromefoundation.org. And if you're ready to start the process, please fill out that form. I actually filled that form this morning, so I'm looking forward to possibly going to Baltimore with my son and maybe making a little vacation out of it.